Okay, since you got such a break yesterday, I'm going to reclaim 2 minutes. Since you're all here, go ahead and get started. So, did you all get your doors all set up? And I know a few of you need oil immersion bottles, so I'll try to track some down. Other than that, we can be pretty good. As we drove back to the memorial service yesterday, we said, you know, we never want to do this again, so we don't want to have any young graduates pass away. Okay. Questions? How many of you have looked over the posted notes for the first set of materials introduction? You should be doing that, okay? You should be reading through those because as I go through that material, then if you've got kind of an idea on what's going to be covered and where we're going, then it's easy to fill in the gaps. And I really would urge you to have those available, and you can use those in the PowerPoint slides for just taking your notes. So you don't have to write down everything. A lot of that stuff is already written down. So take advantage of that particular resource. Okay, next week in lab, we'll do the first lab, which is how to make media. You have to make media one time, and then from then on, we'll provide it for you. But I think it's important to understand how you make your microbiological media, what's in it. Then we'll do a couple of little exercises about the need for aseptic technique in the lab, and do a little bit of microscopy to start to get you introduced to the microscope. So it's a pretty lengthy lab, so it's going to take some time to read through it, so don't leave it until the last minute. Okay? All right, we are kind of going to finish up maybe today, might bleed over to Friday. And by the way, Friday, since we don't have a lab, we'll just have another regular class session. But a little bit of the historical development of the field of microbiology and how we got to where we are, who some of the key players were, what some of the key events were. I mentioned earlier on that there's a period of time kind of called the golden age of microbiology, when microbiology really became established as a science. And that was in the mid-1850s. One of the questions that I really want to dig into a little bit is von Leeuwenhoek, who remember was in the 1600s, first discovered these little tiny, he called them animalcules, tiny animals, in this peppercorn immersion of water. Yeah.
Did you do your test?
I did. I got into folder. I did. Thanks for reminding me. So we've known about, or some people have known about microorganisms since the mid to late 1600s, but the golden age of microbiology, when it really became established as a science, wasn't for almost 200 years later. So why the lag between the discovery of these organisms and the development of the science that studied them? That's what we're going to kind of talk about today. And in the process, we'll introduce you to some interesting concepts. The reason that there was a lag of approximately 200 years between the discovery of microorganisms and the development of microbiology as a science was really a lack of techniques that allowed controlled experiments to be done. Okay, can you have science without the ability to do controlled experiments? No. Okay, that's one of the cornerstones of science. But the ability to do controlled experiments with microbiology was challenging at that time. There were certain fundamental techniques that just weren't around. No one knew how to do them. No one knew how to do sterile technique. No one knew that it was important to do sterile technique or to develop pure cultures of microorganisms. Remember, in controlled experiments, the key is that you can only have a single variable in your experiment. And you don't want to have a mixture of microorganisms, for example, and trying to do an experiment on them, because how can you then correlate an outcome with a particular organism if there's multiple organisms? And yet, we know now that microorganisms are everywhere, and they typically exist in mixed populations. So if I do a culture from my skin, I'm going to get Staphylococcus epidermidis. That's one of your common skin inhabitants. But there's going to be other stuff there too. So if I want to do an experiment with Staphylococcus epidermidis, how do I isolate out that one type of bacteria from all the other so I can then do these controlled studies? Okay? They're so small you can't just reach in there and just pick one out, okay? Like segregating a group of animals into an experimental and control group. So it's a real challenge. Well, what really happened is these techniques emerged over time. It's not like a bunch of people got together and said, okay, we're a dumb science. We can't do controlled experiments. We need to have these particular techniques to be able to do that. The techniques developed as scientists of the time were investigating other questions, and as those questions were investigated, these techniques arose and developed out of those particular inquiries. And then once we had those techniques available, which were aseptic technique, which is basically sterile technique, and then the ability to produce pure cultures, which is a single organism growing in an environment not contaminated by any other organism. Once we had those techniques, then microbiology was free to kind of develop as a legitimate science. Does that make sense? So there were two major questions that the fathers of microbiology, if you like, were investigating for a couple of hundred years that ultimately led to the emergence of these techniques, which then were the foundation of the development of science of microbiology. What were the two questions? One, does spontaneous generation occur? That was one big question. The second question that was being investigated, kind of parallel to this really, was what causes contagious disease? Some people, like Louis Pasteur, who is one of the giants of microbiology, was actually involved in investigating both of these questions. So let's take this one first, and we'll see how this particular investigation led to the understanding that it was important to do sterile technique, and then some techniques developed that allowed that to happen. Another word for spontaneous generation is abiogenesis. We know what that means? What does A mean with that? A mean without. Yeah, life coming from non-living things. According to the concept of spontaneous generation, non-living materials could give rise to living organisms. Now that seems kind of bizarre for us right now because we're firmly grounded in the concept of biogenesis. Life comes from life, right? Why would anyone believe in abiogenesis? Well, you saw a pile of rags in the corner of the hovel you were living in. And all of a sudden, mice emerged from that pile of rags. Yeah, thinking wasn't real sophisticated at the time, but that was a conclusion that was oftentimes drawn, was that pile of rags was giving rise to a living organism. Or what about the meat that had been sitting around unrefrigerated, that all of a sudden sprouted maggots? The non-living material, the dead meat tissue, was giving rise to this living material. And this belief was really grounded in this idea of some kind of a supernatural force which could kind of imbue life into non-living things. And some of the biggest proponents of spontaneous generation were clergy, because this really then kind of invoked this godlike driving force that was responsible for creating life. Okay? Well, some interesting, fairly simplistic controlled experiments were done that pretty quickly put the nail in the coffin on spontaneous generation at the macroorganism level. So Francisco Redi, for example, did this very simple experiment. His hypothesis was that maggots were coming from flies. And now that we know something about the life cycle of insects, we know that the flies lay eggs, eggs develop into larvae, and maggots are the larval stage of the fly life cycle. So what did he do? He took some meat, and he left some in an open container, and he left some in a container that was sealed off with cheesecloth to prevent the flies from getting to the meat. And he observed them. And what did he see? Maggots developed in the open container. They did not develop in the sealed container. Okay, so a very simple experiment that was supportive of his hypothesis that the life, the maggots, were coming from other living organisms, not from the not living unit. And there were other similar experiments that were done, and they just accumulated, and the evidence really accumulated that spontaneous generation isn't producing these complex organisms. I mean, the rats aren't coming from the rags, the maggots aren't coming from the dead meat. But where it was a little bit harder to disprove was at the microorganism level. If you read any history about the people who were proponents of spontaneous generation, they were a tenacious lot. Boy, they just hung on to this belief because in part it was grounded in their faith in the supernatural creator. And they just hung on to this belief and said, well, if it's not happening at the macroorganism level, what about situations like where you have a sealed container of broth? Okay, broth can be made by making infusions of plant material like hay. You soak some hay in water and material leaches out of the hay. Or you boil meat and make a broth from it. And then you take out all the chunks and you reboil it again to kill anything that might have persisted in the broth. And then you observe it. And over time, what happens is that solution becomes cloudy, where it was clear when it was originally produced. And if you take a little drop of this and look at it under a microscope, it's teeming with bacteria. Okay? So we've got this, what was originally, they thought, a sterile broth that they started out with. And then they let it sit for a while at some appropriate temperature. And all of a sudden, living organisms appeared. And they said, spontaneous generation, right? Okay. Many people disagreed with that particular idea and instead believed that dust from the air was settling into these broth infusions and that there were bacterial cells adhering to that dust and they were being deposited in the broth, and then when the broth was sitting for a while, especially if it was the right temperature, those cells started dividing by binary fission and we started to form a cloudy solution. Or a turbid tube resulted. Okay. So some early experiments were done. This is an experiment by a guy, I can't talk, by Chabot, where he did some very simple experiments where he took broth solutions that had been boiled, and so hopefully they didn't have any living cells left in them. And then he covered some and he left some open, and he found that the covered ones had no microbial growth. The uncovered ones had heavier microbial growth, kind of a similar idea to what Rady did at the microorganism level, the macroorganism level. And this seemed to be supportive of the idea that contaminants from the air were causing the growth to occur. Life was giving rise to life, not abiogenesis. The experiments really became a little bit more sophisticated over time. And Schultz and Swan, for example, did an interesting experiment where they took some previously sterilized infusions and put little glass tubes in them that allowed fresh air to come into the tubes. But they also had the tubes coiled, and they used a little flame of an alcohol burner to sterilize the air that might pass through this as it's going into these vials. And When they set this particular system up, they found out that these particular sterilized infusions remained sterile. The person who really put the nail in the coffin of spontaneous generation was Louis Pasteur. And he was in the mid-1800s. He was one of the golden age of microbiology people. Pasteur did a very simple and elegant experiment that I'll show you in just a second that really did refute the hypothesis of spontaneous generation. He had other things that he did as well. He developed the technique of pasteurization, which we'll talk about later. He was a very important contributor also to the germ theory of disease. Now, Pasteur was also involved in this investigation of the second question: what causes contagious disease? Here's what Pasteur did. He worked with flasks that were called swan neck flasks. When they made the flasks, they were sealed, and they drew the glass as it was blowing out into this long neck-like structure with a bend in it. Okay? So it looked like the neck of a swan. He took broths and boiled them to destroy any contaminating cells. Others he didn't boil. And what he found out is that once you had boiled the infusions in these sealed flasks, they remained sterile. They didn't develop that cloudy turbidity. And if he took this particular flask that was sterile and then broke the neck so that contaminants could now get inside, it got cloudy. Or if he tipped the flask so that the liquid went into the bends down here where microorganisms might be able to enter, and then tipped it back again so that contamination got inside, that flask became cloudy. There were flasks that were done in the 1800s that he prepared, which are now in the Pasteur Institute in Paris, which are still completely clear and microbial free after a couple of hundred years. Okay? So once you achieve a sterile situation in here, if it's not exposed to any external environmental contaminants, those particular broths remain sterile pretty much indefinitely. Okay? Now, this is pretty much what Schultz and Swan did. Why was Pasteur's work so compelling and convincing where Schultz and Swan's was just kind of disregarded? Because Pasteur had the credits. Okay? Prior to doing this, Pasteur was credited with saving the French wine industry. And the wine industry in France at that time was huge. Okay? The French wine industry was in real trouble because all of a sudden, all of the winemakers in France were having problems with their wine becoming sour. And that's not good. What Pasteur believed was that there were contaminating microorganisms, particularly yeast, which were growing on the skins of the grapes. And those yeasts, contaminating bacteria, which were growing on the skins of the grapes. The yeast you want on the skins of the grape because when you make your wine, they ferment the sugar and produce alcohol. But the bacteria were fermenting the sugar and producing lactic acid. And that acid caused the wine to sour. So he tried to figure out a way where he could selectively destroy the bacteria, but not kill the yeast, prior to the fermentation process, and then we would get alcohol production and not acid production. So he developed this technique where you mildly heated the liquid, and he found just the right temperature that would kill the bacteria, but would not kill the yeast. And that process of selectively destroying bacteria through differential heating was called pasteurization. So he really had the creds, and then when he did his spontaneous generation experiment, everyone went, oh, okay, Louis Pasteur, we know this guy, we believe what he had to say. He later went on and developed a vaccine for rabies. As I said, he was involved in the germ theory of disease. He did a lot of study about microorganisms and their ability to cause spoilage through fermentation. So this is really one of the giants of microbiology. So what arose from these kind of investigations of spontaneous generation? Well, number one, there was this realization that there are microorganisms out there that can contaminate things. And that you can kill those microorganisms if you heat them enough. Now, there were some little wrinkles in this realization because what they found was that boiling was not always sufficient to kill all the growth in a broth, for example. Sometimes you'd boil a broth and put it in an enclosed flask, and it would develop growth again. But what's going on here? Other situations with different kind of broths, you'd boil it and remain clear. Well, what they eventually figured out is that some bacteria can produce highly resistant structures called endospores. And endospores are not killed by boiling. So one of the techniques that developed during this particular time were methods of sterilization that would kill not only cells, we call them vegetative cells, but would also kill the endospores as well. Do you know what technique I'm talking about? How do you make sure that something's completely sterile? It doesn't have any vegetative cells or any endospores that could give rise to vegetative cells. You've done it. You've done it. Autoclaving. Okay. Autoclaving is basically pressure cooking. And what's the theory behind autoclaving? Why will autoclaving kill something when boiling won't? It is pressurized. Okay. Typically autoclaving is done at 15 pounds per square inch pressure. Is it the pressure that kills the microorganisms? No. What does pressure do to liquids? You ever taken the radiator cap off your car when it's hot? What all happens to your coolant? Right? Increased pressure elevates the boiling point of water. So instead of water boiling at 100 degrees centigrade at atmospheric pressure, at 15 pounds per square inch, it boils at 121 degrees centigrade. So we can achieve a higher temperature within the autoclave, and that extra 21 degrees centigrade is enough to kill the endospores. Okay? So the techniques of autoclaving, ability to kill microorganisms by sterilization with heat. We're going to be flaming loops that we use to transfer microorganisms using Bunsen burners. All of those kind of techniques grew out of this understanding that there's contaminants everywhere, and if we want to be able to develop pure cultures of microorganisms, a single species, you've got to be able to eliminate the contaminants. Okay? So we really owe the development of aseptic technique to this phenomena of trying to answer this question of whether spontaneous generation was really occurring. Okay? So, second question. What causes contagious disease? Well, even as far back as the 1300s and the 1400s in Europe, it didn't take a bright person to know that diseases could be passed from one individual to another. Because that was the time of the Black Plague. Black Plague, which turned out to be a microbial infection, affected about 2/3 of the population of Europe at the time. And half the people died. And you looked around and you saw that if one case of plague developed in your city, there were soon to be many other people that were going to get sick. So what was causing this? Okay, well, this was well before von Leeuwenhoek's time. And nobody knew about these little animalcules. No one knew about bacteria. And so there was all this interesting ideas about miasmas literally means bad air. Somehow there are these evil particles that exist in the air that if you breathe them in can cause people to become sick. Sometimes people notice that if you're around corpses of people who had died, that you could then get sick. And so there was a theory about cadaveric particles that were causing the illness. That's one of the reasons why the practice of burning corpses became fairly well established pretty early on. Just incinerate those suckers so they can't release their cadaveric particles. But it took a long time for people to nail down a cause and effect relationship between germs and diseases. When we talk about germs, we're talking about primarily bacteria, but you have to throw virus into that particular category because they can cause disease. And you also have to throw fungi into that particular category as well. The person who is credited with the development of the germ theory of disease is Robert Koch. And again, Koch lived in the mid-1800s to early 1900s. But it took a long time for this particular germ theory of disease to develop. Along the way, there were some kind of circumstantial pieces of evidence that hinted at that particular relationship. But it really wasn't until Koch did his seminal work that a cause and effect relationship was established. So, let's talk a little bit about some of the people who accumulated some circumstantial evidence for the germ theory of disease, but who never really quite put it together. One person was a physician by the name of Semmelweis, and he did his work in the mid-1800s. Semmelweis ran a hospital, and he was particularly concerned that there was a very high incidence of childbed fever. And childbed fever was a disease that newborns oftentimes got. And infants died very quickly if they contracted this particular disease. He had a hypothesis. He believed that microorganisms were being transferred from physicians, particularly those that went into the operating room or worked with corpses first, and who then went and tended to maternity wards and exposed young children to these particular pathogens. So he established a process of hand washing and required all of his physicians to wash their hands in between visiting patients. And he used a chloride compound. Chloride's lime. And lime can be kind of antibacterial because of its basic pH. And we'll see later on that chlorine compounds are pretty effective antibacterial agents. As soon as the physician started washing their hands, incidence of childbed fever really plummeted. Okay? And he ran into a lot of opposition. And ultimately, he was ridiculed by a lot of the doctors who didn't want to comply, forced to leave the hospital. He became depressed, went into a sanatorium, and died there from a staph infection. Okay. So people really didn't want to believe this, and doctors in particular didn't want to be told what to do. In 1853, something else interesting happened. A British physician, John Snow, not the Game of Thrones person, was working in London, and there was an outbreak of cholera. which we now know as a bacterial waterborne disease. And people were dying left and right. Snow started to correlate and map where the outbreaks of cholera were occurring in London. And literally, he was putting little pins in a map to see where the outbreaks were. He really went around and visited all the apartments and things and assessed whether there were sick people there or not. And he started to see clusters. And one of the things that he saw was that most of the clusters were centered around a commonly shared town pump. And as it turns out, that town pump had been contaminated with sewage. And the bacteria were then getting into the water, and people were drinking the water. And so I don't know if this is really true, but according to legend, he removed the handle from the town pump so people couldn't use it, and the cholera epidemic stopped. He's credited with conducting one of the first epidemiological studies. We'll talk about epidemiology, but that's where you try to correlate outbreaks of disease with particular locations and try to find causes associated with that. Pasteur was involved in showing that bacteria were responsible for souring wine. Already mentioned that. In 1867, another physician, Joseph Lister, became really concerned about the incidence of surgical infections that were occurring in his hospital. So he instituted a procedure where they did sterilization of surgical instruments. And instead of just operating on someone and then wiping off your material, your instruments with cloth, for example, and then going and operating on the next person, he mandated that the instruments be treated with carbolic acid, which is also known as phenol. And we now know that phenol is a very effective antibacterial agent. It denatures proteins very readily and kills cells. And again, when he instituted this particular mandate, the incidence of surgical infections plummeted tremendously. So there were various people here and there who had some kind of inklings that these microorganisms were causing disease, but there really wasn't any correlation that could be pinned down between a particular microorganism and a particular disease. And that's where Koch came into play. Okay? Koch was a person who was credited with the first demonstration of a cause and effect relationship between a particular disease and a particular microorganism. As Koch carried out these particular studies, he developed these techniques of pure culture. The ability to grow a microorganism in isolation from all other microorganisms. It didn't have any other contaminants associated with it. Koch was working with a disease that didn't primarily infect humans, but instead infected mainly cattle. And the disease was anthrax. You heard of anthrax? No? You heard of it? Have you heard of it? Yeah. Once a herd became infected with anthrax, you pretty much had to sacrifice your whole herd because it was going to spread like wildfire. And if you didn't, then it was going to likely spread to other herds as well. Koch did some initial studies on the blood of cows infected with anthrax, and every time that he looked at the blood of a sick cow, he saw in the blood rod-shaped or bacillus bacteria. And when he looked at the blood of healthy cows, that bacillus bacteria was not present. So is that definitive evidence that the bacteria are causing the disease? correlation, right? Was correlation always causation? Is it possible that the bacteria are thriving because of some condition that the disease causes? The disease could have come first, right? Okay. Koch did another interesting set of experiments. This must have taken a lot of cattle to do this. He would take a diseased cow and draw some blood and inject it into a healthy cow. And that would get sick. He would then take that newly sick cow and draw blood and inject it into another healthy cow. And that got sick. That was called making passes of blood from one organism to another. And he did that for up to 20 cows. And every time the newly injected one got sick. That suggested to him that the disease of anthrax was caused by a living organism rather than a toxin. Why? Why does that particular observation suggest a living organism rather than a toxin?
If it was a toxin, it would be like diluted every time.
Absolutely. If it was a toxin, it would be diluted as it was passed through the bloodstream of successive cows. Okay? And every time he looked at those particular newly diseased cows and looked at their blood, they had these bacillus bacteria. Well, this was pretty compelling evidence. The problem, though, and the criticism that some of his scientific colleagues levied was, you're injecting this blood, and you're assuming that what you're injecting from the blood, the bacteria, are causing the disease. So could there be other things in the blood besides the bacillus bacteria that might be causing the disease? And he had to say, well, it's a possibility. I haven't accounted for that. He really hadn't properly controlled his experiment. So what he really wanted to do was to be able to make a pure culture of that bacillus bacteria. Have a sterile solution that only had bacillus growing in it. And then, instead of injecting blood into a healthy cow, inject that solution of a pure culture of bacteria. And then, if the cow became ill, you could correlate it with the injection of that one particular thing. Okay? But he didn't know how to make a pure culture. Okay? How do you separate out the bacillus from this blood from this infected cow? Well, he tried several things. He tried a technique called selective enrichment. And basically what he did was he took some blood and put it in some growth media and hoped that the growth media would favor the growth of the bacillus bacteria, but not favor the growth of anything else. So the idea was is if this blood had, let's use X's and zeros as different species of bacteria, that this media would favor the growth of X's and not favor the growth of zeros. But try as he might, there were always a few that survived. He then tried the opposite kind of idea of selective destruction. Could he create a media that would selectively destroy one thing but not another? And again, he could get an enrichment of a species, but he could never get absolutely a pure culture. So he's kind of stymied. Well, sometimes things that happen in science happen as a result of accident. Koch had effectively a postdoc working in his lab by the name of Hess. And Hess liked to eat slices of raw potato. Take a potato and slice it off, maybe put a little salt on it, eat the potato in the lab, and drove Koch crazy because he didn't want to have food in his lab. And Hess was also kind of messy, and sometimes he would leave his potato slices laying around. And one day he walked into the lab and realized he'd had some potato slices sitting there for several days, and he was about ready to throw them out before Koch saw them. And he looked at the potato slices, and he saw these little colored dots on the surface. And as it turned out, those little dots were colonies of bacteria. And if you took some material from that colony and you looked at it under the microscope, they were all the same type of organism. Well, where did they come from? They came from airborne contaminants that fell on the surface of the potato. They could grow on the potato, and one cell divided to form two, two to four, etc. And this visible mass of cells was all a clone of that original cell. That colony was a pure culture. So he thought for a second and he said, what if I take a sterile glass plate that one of my colleagues has developed, Robert Petrie dish, and put some potato slices in there and sterilize them, and then take some blood and smear it on the surface of the potato, and hopefully as I do that, cells will come off of my loop that I'm using and be transported onto the surface of the potato, and eventually I'll get a cell isolated from another. You're basically doing a dilution on the surface of this potato. And that a cell could then grow into a pure culture, and then I could pick off that particular colony and put it in more sterile growth media, and then I would have a pure culture. And he played around with techniques for getting good dilution on the surface of this potato. This is something you're going to do in a couple weeks. We do something called streak plating, where you streak some growth on the surface of media, and then you streak again, and then you streak again, and you streak again. And at some point in time, you're depositing single cells onto the surface of that agar or potato, in this case. And then those are growing and giving rise to clones or colonies of pure cultures. Problem is that the bacillus bacteria didn't grow on potato. Potato was really a pretty lousy nutritive media for growth. So we started playing around and saying, are there other things that I could use as my growth media besides potato? And one of the things that he tried next was gelatin. Gelatin's got protein in it, pretty good growth media. So he would make these plates of gelatin, let them solidify after they've sterilized, streak out the growth. These bacillus bacteria preferred a fairly warm environment, so we put them in a warm incubator, and the gelatin would melt. Okay, because gelatin melts at elevated temperatures. So gelatin can be used as growth media for some microorganisms that have lower temperature optimum, but not these bacillus bacteria. Well, they were getting kind of frustrated, and then Hess was talking to his wife, who was of Caribbean origin, about this problem, and she said, well, you know, when I cook, I use a lot of this seaweed derivative called agar. And this agar stuff seems to be pretty nutritive. It will dissolve if you boil it, but then when you let it solidify at room temperature, if you put it in an incubator, it will stay solid. So what he was able to do then is prepare these sterile Petri plates of agar, let them solidify, take his blood from the diseased cows, streak it for isolation onto the surface of this media, and get pure isolated colonies, which then he grew up in more agar, and he had a pure culture of microorganisms. Now when he injected those into healthy cows, they got sick. He was able to observe the bacillus bacteria that weren't there before, and so the appearance of that bacteria was clearly correlated with that particular act of injecting that pure culture of bacteria. This particular process is now known as Koch's postulates, and Koch's postulates are the gold standard for correlating particular diseases with particular microorganisms. So basically you have to have a pure culture, you have to be able to isolate that outside of the body, inject it into healthy animals. Healthy animals should get diseased, and then you should be able to re-isolate the pathogen from the newly diseased animal, and it should be identical to the one that you injected originally. And that series of steps, Koch's postulates, is still used today by the Center for Disease Control anytime there's a new disease that they're trying to correlate with a new particular infectious agent. It becomes a little bit more challenging with virus because it's difficult to grow a virus outside of the human body and get pure cultures. So with virus, you have to do human tissue culture techniques to grow the viral pathogens. Well, once the relationship between microorganisms and diseases was finally established, people really started to take interest in microbiology. And a lot of the original kind of fathers of microbiology were physicians who were looking at trying to identify causative agents of particular diseases, tuberculosis, cholera, smallpox, you name it. So the interest in microbiology really grew, and now that we had this ability to do controlled experiments because we had the proper aseptic techniques and pure culture techniques, microbiology really took off as a science. This slide kind of gives you a little bit of historical context about some of the people that were developed in kind of this early establishment of microbiology. And microbiology is now grown to this incredibly important science that involves immunology and gut bacteria and genetic engineering. Microbiology is really one of the hot areas right now. And it's a relatively young science. It really dates its validity really back to the mid-1800s, but the foundations of that were laid for a couple of hundred years prior to that. So, we've covered the who, what, why, where, when. It's kind of a original context for why we're going to spend our semester looking into these prokaryotic organisms. So what we need to do now, next time, is start talking about the differences between prokaryotic and eukaryotic cells and bacterial structure. Okay? As I said before, prokaryotic cells are pretty simple. They don't have all of those membrane-bound organelles. But there are structures that are species-specific that are found in some bacteria, not others. For example, not all bacteria have endospores. Okay? Not all bacteria have flagella. There are various storage granules that are found in some species of bacteria and not others. There are fundamental differences in cell wall structure of two major groups of bacteria, the gram-positive and the gram-negative bacteria. So the next topic we'll talk about will be the kind of basic bacterial structure. And that's immediately going to lead into applications in lab. Because one of the first things you're going to learn in lab after you learn your sterile technique is how do you do stains to determine whether your bacteria is gram-positive or gram-negative? And what does that tell you about your bacteria? How can you detect the presence of flagella? How can you detect the presence of a PHB granule? And so there's going to be a real correlation between what we talk about in class in terms of structural characteristics and then what we're looking for in lab. So I would really urge you to look through the section prior to tomorrow, at least the beginning few pages of the bacterial structure lecture notes. And then over the weekend, I'm going to look on mastering. And I know there are some relevant materials there that I'll call your attention to, particularly in regard to cell wall structure and some of the other more complex aspects of bacterial structure. So that's where we're going next. Okay? So I let you out early when I started early. That's just wrong. Okay? See you tomorrow. Okay.